Today, we are very excited to have with us Dr. Jeffrey Millman at Washington University, St. Louis, WashU. Um, his talk is identification of the unique cell types responses in pancreatic islets to stress. His new paper just came out recently. I'm going to drop in the chat in a minute, and it's very exciting. It's very methodical, very detailed, and it's the first attempt to really qualify and quantify what is going on in the human stem cell islets when they're exposed to different stressors. So just a little bit about him. He is an associate professor of medicine and biomedical engineering at Washington University of Medicine. He received his PhD from MIT and completed his postdoctoral training at Harvard. His current research is focusing on synergizing both biomedical engineering and cell biology approaches to use stem cells for study and treatment of diabetes. And he's developing new um, approaches for the differentiation of insulin-producing islets from stem cells and improving their utility for cell replacement therapy and in vitro disease models. His innovations have been licensed to biotechnologies companies in the diabetes space and resulted in ongoing clinical trials. Dr. Millman previously served as the Vice President of Research at Sana Biotechnology, where he oversaw the preclinical development of hypoimmune stem cell-derived islets for the treatment of type 1 diabetes, and his laboratory is supported by the NIH, NIDDK, and JDRF, and his scholarship has recently been recognized by awards from JDRF, American Institute of Chemical Engineering, and the Biomedical Engineering Society. Thank you so much for joining us. I can't wait for you to walk us through this paper.
Yeah, well, I really appreciate the kind introduction. Let's see if I can do the incantation to share my slides correctly.
I'm going to
That
drop
is probably
like that.
flipped, right? Let's do
Yeah.
that.
Good. Also, you might want to do full screen. That's perfect. And if anyone has a question, please drop them in the chat as we go.
Great. Thank you very much. Before I get started, let me hide this real quick so I can actually see my slides. There we go. My university requires me to do some disclosures about some conflicts I have regarding patents and other relationships with companies, which are outlined here. There we go. This is likely very familiar to people of this audience, but the pancreatic beta cell is central to diabetes, where the cause of diabetes across all types is ultimately there just not being enough healthy beta cells to keep up with the metabolic demand of patients. And the reason for this loss of healthy beta cell mass is the combination of many different factors, such as immune cell attacks or environmental factors or genetics as well, that work in a way that we don't fully understand to cause there to be a insufficient number of healthy beta cells in order to maintain normal glycemia. Fortunately, we have insulin injections that can manage this disease, but it's a very imperfect treatment with patients having persistent issues of lack of blood glucose control that results in a increased likelihood of death, decreased lifespan, and also many other associated difficulties in their health and the actual management. Fortunately, we do have a cell therapy that is effective in managing diabetes. This is the transplantation of islets containing the insulin-producing beta cell that can be given to patients to avoid the need for insulin injections to manage their blood sugar levels. Though, and this is just some data here showing from a review article from some colleagues of mine a few years ago now, showing how effective this can be in a patient with type 1 diabetes. Of course, similar to the underlying issues that gave rise to diabetes in patients to begin with, transplanted islets can also be subjected to the same sort of stresses and environments that cause the diabetes to form in the first place, which could have major implications in the long-term stability of this cell replacement therapy. Another issue that cell replacement therapy faces is the large number of people worldwide who are living with diabetes. and in order to be able to supply even a fraction of these patients with the cells to give them the option for a cell therapy, we would need a large-scale sustainable source of replacement cells and tissues in order to meet this demand. And so a major focus in my laboratory is the generation of stem cell derived islets in the laboratory, utilizing the ability of stem cells to undergo a process known as self-renewal. That is, that they can proliferate and divide and make more of themselves if given one set of cues, or given a different set of cues. They're capable of going through a process known as differentiation to differentiate into potentially any cell type found in the body if you give them the correct combination of cues to do so. Obviously, we're mostly focused on beta cells and pancreatic islets. And we've published several reports showcasing improving ways of actually making these cells in the laboratory, which look like, again, these are completely artificial lab-grown cells, but they at least superficially look like pancreatic islets, for which many of the cells are capable of producing insulin. So I'm not going to go through a lot of the details of that because this is a relatively small part of this actual paper here, but just to showcase what these cells are capable of doing, they're able to very quickly reverse diabetes in severe hyperglycemic mice. However, these cells are also quite immature, and we're always looking for ways to improve them. So the main thing I wanted to talk about here today, with that background being recognized, is that as part of our efforts in trying to study both stem cell derived islets and primary islets, we've been investing quite heavily over the last 6 years or so into using single cell sequencing methods in order to gain new insights into the biology of these cells. And the reason why single cell sequencing is particularly important for the islet is because of what I like to call the smoothie effect, that conventional assays such as real-time PCR or Western blots are kind of like investigating a smoothie, which can be quite delicious. You can learn a lot from drinking a smoothie in terms of guessing about what's in there, but you obviously lack the specificity about the details of the fruit that went into the smoothie, such as the the color of it, the taste of the individual pieces of fruit. And so the analogy here is that for a lot of conventional assays studying islets because of their heterogeneous nature, we're looking at islet smoothies here, where we're looking at and we cannot necessarily, with these bulk assays, we cannot necessarily assign any particular readout there specifically to the beta cell versus the alpha cell versus perhaps exocrine cells that are also part of that islet isolation. And again, this is very important because, particularly in the case of type 1 diabetes, it's a disease that seems to be very specific to a autoimmune attack against the beta cell that doesn't occur for the other cell types there. And fundamentally, we don't understand why that is occurring in this disease. But you can imagine kind of more broadly here that these single cell assays can be quite important in answering many questions of interest to both bioengineers that are trying to make stem cell derived islets for cell therapy, but also more islet biologists that are trying to understand the mechanisms of disease and perhaps trying to design a pharmaceutical intervention that could protect the beta cell from a environmental stress or a immune attack. And so this study that I'm going to talk about here in a few minutes was really motivated by our initial studies just studying both stem cell derived islets and primary islets under unstressed sort of conditions. We were doing this initially to both make sure that the single cell approach could get us new insights and to lay the foundation for us to do more complicated studies such as the stress response of cells. But just give a little bit of background about how both stem cell derived islets and primary islets look under kind of basal conditions here. In the study that we published last year, we used single cell multiomics sequencing to get the transcriptional identity and the epigenetic identity of each of the cell types found in stem cell derived islets and primary islets. And using these sequencing methods and referencing to markers of cellular identity, we're able to identify the usual suspects in both stem cell-derived islets and primary islets, including obviously the pancreatic beta cell, alpha cell, delta cell. We get not a whole lot of other non-endocrine in the stem cell differentiations here on the left. You can see a little bit of exocrine and a little bit of mesenchyme. The primary cells have more cell populations that we can identify, typically more exocrine impurities that are part of this, some endothelial cells, some immune cells as well. But one of the great things about this is that we can start going and doing a head-to-head comparison, basically eliminating, by using single cell methods here, we're able to eliminate the potential of essentially dilution effects of other cell types that are not of interest to that comparison. In other words, we're able to essentially computationally purify, and for this example I'm going to be showing here, compare directly just the beta cells from, in this case, stem cell derived islets versus primary islets. And there's obviously a lot of similarities in these. I'm just showing just the beta cell data here right now to illustrate this technology that we're able to see here a lot of similarities between SC beta cells and primary beta cells, but I'm emphasizing the differences here because that's more interesting to talk about, I think, with the beta cells typically, the primary beta cells typically having a greater amount of RNA expression for markers considered to be maturation markers of beta cells, such as insulin itself, IEPP, G6BC2, UCN3, and MAFA. But also we can see this maturation reflected in the epigenetic identity of these cells. So here I'm showing the chromatin accessibility of transcription factor DNA binding motif. So these are the regions of the DNA that transcription factors are able to recognize and adhere to do their biological function. If the chromatin is closed for these binding motifs, then the transcription factor can't come in and bind and regulate transcription. However, the chromatin is open and the transcription factor is there, then the transcription factor can come in and bind and regulate transcription. So you need both RNA expression and also chromatin in order for transcription factors to do their biological function in gene regulation. And here we can also see that it's not just RNA expression, but it's also the epigenetics of the cell that illustrates stem cell-derived beta cells as having being in an immature state, with some of the more interesting aspects of this being that stem cell-derived beta cells do have open chromatin for off-target identities, such as progenitor identities and intestinal identity. And I'll skip the MAFA thing for now. This also can be seen not just focusing on chromatin here for just a moment. This can be seen not just in the DNA binding motifs for transcription factors, but also in the chromatin for the promoters themselves. Obviously, you want to have open chromatin for the promoter for a gene in order for transcription factors to come in and regulate the expression of that gene. So here I'm showing both on and off target promoters, comparing primary cells to stem cell derived cells. There's a lot of cell types here, I realized. So I went ahead and drew a little red box just to point out what the beta cells are, because that's probably what most people are interested in on the call here. And you can see that with the primary beta cell, that the promoters are open for common beta cell markers and are mostly closed for markers of other endocrine identities such as glucagon. However, this distinctness is not seen as well in the stem cell derived tissues, with the SC beta cells typically having moderately open promoters for both on and off target identities. This is true for both the endocrine genes that I'm showing with the blue panel, but also for non-endocrine identities as well, which is with the orangish colored panel on the right here, where you can see that the beta cells will have open promoter accessibility for non-endocrine off targets such as exocrine and also progenitors. So this is where we're starting at, where we were really able to see major differences in the maturation state of stem cell derived islets versus primary islets using this technology. And so we really wanted to bring this technology back to answering more fundamental questions about how do islet endocrine cell types, and I guess exocrine cell types as well, we'll talk about here in a moment, respond to stress. Obviously, there's been a lot of papers over several decades at this point showing that islets can respond to various stresses that are found inside of the body, but due to technological limitations, have been really difficult to ascribe specific responses to stresses to the beta cell versus other cell types in the isolated islets. And so here we wanted to use, again, the single cell multi-omic sequencing to answer this question: how do specific islet cell types respond to stress? And so here at WashU, we collaborated with Dr. Fumihiko Urano, who is an investigator here in the Division of Endocrinology and also the Diabetes Research Center, he's also an expert in islet stress, to use our technological platform to answer this question. The first author of this paper is Molly Maestas, who's a fifth-year PhD student and will be on the job market next year, looking for a biotech job for anybody on the call who's interested in that. And also several current and past members of the Millman lab who are listed here on the bottom. And so with that background out of the way, here is the basic design of the experiment that we had in this paper that was published in Nature Communications a couple of months ago at this point, where we received isolated islets from 6 deceased donors. So these are non-diabetic donors, and we were not trying to get patients of a particular demographic either. So the demographics are a bit random here and not really powered enough for us to make any statements about that really. But we're able to get 6 donors from Proto Labs, culture them ex vivo for 2 days under several different culture conditions that mimic aspects that have been, or mimic environmental factors that have been identified in diabetes, including BFA, which can inhibit transport proteins from the ER to the Golgi, thapsigargan, which inhibits calcium transport to the ER, causing ER stress, and various different combinations of inflammatory cytokines. And then, so we were able to culture these islets under all these conditions, and then apply various different sequencing methods to them. Basically, as soon as we got better tools, we started using the newer sequencing methods here. So this is really kind of representing the evolution in my lab of being able to do this sort of work. And mostly focus on trying to multiplex these conditions in order to make this experiment fiscally realistic for us to be able to do. All in all, again, we had the 6 donors here. We sequenced over 133,000 cells, and we used the sequencing data to define cell type identities and the specific responses to these diabetic stress. So in addition to the new data that we generated in this paper, and this is all deposited on Geo and freely accessible, we also compared it to other published sources of data, including data from the HPAP database of patients with type 1 diabetes and are also autoantibody positive, and also to a non-diabetic paper that was published a good amount of time ago from a different team. And so for this, particularly this first method, which is where we did most of the analysis for the figure, or for the figures in the manuscript, we would take these isolated islets, subject them to these different stresses, could use a antibody with a DNA barcode on them to barcode the cells so we can mix them together in order to sequence them all together. And then because we knew what the sequence of the barcodes were, we're able to computationally demultiplex these pooled samples together in order to figure out what's the to correlate the sequencing results to the treatment that we were that we had subjected the cells to. And so here is for three of the patients with this DNA-based multiplexing approach, what we were able to see in terms of cell type identification. Probably one of the more obvious nuances here is the fact that we can identify multiple different types of beta cell and alpha cell populations that on this two-dimensional projection are quite a bit different from each other. And again, you could probably guess this is because of the treatment group, and indeed that's the case. Going back to the DNA barcoding, we're able to figure out what conditions gave rise to which beta cell or alpha cell subtype here, and correlate those together. So you can see, just to guide you here a little bit. So this is beta cells 1 population, includes the control and some of the, I guess, weaker stressors here. The beta cell 3 is predominantly the BFA treatment, and then beta cell 2 is predominantly the cytokine conditions that had interferon gamma in them. And there's a similar trends to be seen with the alpha cells as well. And just to kind of, as a bit of a sanity check here, we did look at some of the common known transcriptional responses to these stresses, and we're able to see a decent correlation there. Maybe CXCL10 is one of the easier ones to see on this slide here, where you have high expression of it for the alpha cell and beta cell populations that were treated with the cytokine mix. So one of the quality controls that we did was to compare our data with published data sets. And so in this case, we were looking at the first three patients, looking for the control samples here, not given any stress, cross-reference it to the 2018 diabetes article, and saw a really good correlation in the alpha cells and beta cells and other cell types from our data set compared to their data set. Another quality control that we did was to make sure there was reasonable consistency across patients, showing the first three patients here. And there is obviously some degree of patient variation, which this paper is not powered enough to really understand fully here, but in general, the trends were the same. The patients were responding in a overall similar manner to the stresses that we were giving them. So we're not giving completely irreducible data, at least. We also looked at, again, the HPAP database here for at least comparing our cytokine mix treated islets to their type 1 diabetic and autoantibody positive beta cell populations, just seeing that gene sets that are commonly associated with diabetes were indeed enriched in both our cytokine treated cells along with the type 1 and autoantibody positive patient samples that they had deposited on their server. So with that quality control out of the way, we started asking some of the, interrogating the data, asking some questions that we had, thought were the most interesting to look at here. One of the dangers of a experiment like this is that there is almost infinite number of ways you can slice and dice the data, and just way too many interesting observations and genes and pathways to follow up on than are feasible. And so one of the things that we spent a lot of time discussing among my team was, what are the most interesting questions to pursue and focus on when we were writing up the manuscript. And so when I told when I would go to conferences and speak to other faculty members about the fact that we're doing this sort of experiment, one of the first questions I pretty much universally got from other people in the field was, oh, do the beta cells respond more strongly in terms of the number of genes that were changed? Because that was kind of an obvious thing that beta cells are maybe more sensitive and would have just a stronger response to a stress compared against the other cell types. Maybe that could be a mechanism for why beta cells are the ones failing in diabetes. And so that's one of the things that we first looked at here. This is just graphing number of differentially expressed genes for all the cell types across all the stressors here. And there's obviously some differences in the number of in how specific cell types respond, but the beta cells were not unique in terms of how many genes were being differentially expressed. In fact, it seemed that if you were to do a head-to-head comparison here, the alpha cells actually express a few, for most conditions, express more differentially express more genes than the beta cell. And overall, we saw that the alpha, beta, and ductal cells, surprising to us, were seem to be the most sensitive to the stresses from this kind of simple-minded DEG sort of analysis, with the delta, PP, and acido cells being, able to respond, but not as strongly as those other three cell types were.
Jeff, we
Obviously,
have just two
oh,
questions here. One is, have you
oh,
tried exposing stem cell derived islets to these stresses?
yes. Though we haven't done the same sort of analysis here of doing the like a single cell sort of readout, but the cells are able to respond at least in a similar way to as primary islets were. We chose in this study to focus on the primary islets because we thought that would be more useful to the field given the immaturity that stem cell derived islets have. Though you can imagine a interesting follow-up study for this would be to do the same thing with stem cell derived islets and do a head-to-head comparison to better understand how primary islets and stem cell derived islets differ in their response.
Yeah, good answer. And then someone also asked, I mean, obviously it's not in this paper, but she wanted to know, did you try exposing the islets to other stresses such as oxidative stress, glucotoxicity, and lipoglucotoxicity?
Yeah, for this study, we didn't do that. We spent a lot of time talking about what stresses we should subject the cells to. We were basically limited to the number of conditions we're testing here in order to just get enough cells per condition using this kind of earlier version of the multiplexing technology. In our experience, we had found that these stresses listed more of a response in islets than, well, because we have never really done oxidative stress, but we've done try to do glucotoxicity and lipotoxicity before. We just frankly don't see much of a response in human islets to those stresses, and so we decided not to include those in here and instead focus on several different cytokines and also thapsigargin and BFA.
Great, thank you.
I'm not saying those are bad to look at, but this is just a judgment call. So obviously there's more complexity to this than just like number of genes that are being expressed, and so we did look at genes associated with the normal stress response pathways that have been well described by others in the islets. And this is a lot of data that I'm showing here, mostly to illustrate the immensity of the data set. But I did have a few of these red marks here to kind of illustrate just a few small points I wanted to make. And so this gene up here, HSPA5, better known by its protein name BIP, under control conditions, we identified that the exocrine already have elevated BIP expression even without the stressor. With BFA treatment, we found that the beta cells and ductal cells in particular had the greatest upregulation in BIP. Thapsigargin, again, was on exocrine. But then for cytokine mix, we found beta cells and PP cells, randomly in my mind, were the ones that were most responsive to it. So you can see here that even for this very well studied protein in gene, that different stressors can cause different cell types to respond in their upregulation of BIP differently. But then if you look at other transcription factors that are associated with stress, you'll see different patterns like ATF4 here. Looking at BFA, we now find that the alpha cells and ductal cells are the ones that most upregulate that, which is different than the pattern that we saw in BIP. So we illustrate here the differences in the patterning of the response. We don't fully know what that means in the context of these stress response pathways yet, however. So one thing we were very interested in kind of overall here was understanding the common, sorry, getting my timer here, looking at the what responses were common across cell type and stress and what responses were unique across cell type and stress. And overall found there was a good number of both. there was a good number of unique responses to stress, and there was a good number of shared responses across stress. So there wasn't, and I don't know if that was expected or not. I think we were kind of hoping there will be more, like almost exclusively unique responses, but the data here is a lot more nuanced than we first originally thought here. And I have a few examples down there of some of the more enriched genes that we were seeing. But since we were able to, in this data set, get the beta cell gene expression signature and really looking at genes that were specifically changed in beta cells, we're able to use that gene signature and put this into a drug gene interaction database that's mostly developed for cancer, I believe. But we wanted to see if we could get any drug candidates out of that that could potentially help to alleviate the stress response in a beta cell. And that's some action shots I got of Marley pretending to do work. And so for this, we identified 3 compounds that are listed on the screen here that were hits from the database. And so for this, we turned to stem cell derived islets to see if these compounds could reduce the amount of apoptosis we were seeing with the BFA treatment. And indeed, we're able to identify that these compounds were able to reduce the death of stem cell derived islets under BFA stress. And we chose BFA just because it's the strongest inducer of death that we had out of the compounds we were studying here. And so kind of moving from unique responses to common responses here for a second, we wanted to see if there was what was the most common across cell types here. And so we cross-reference across these, just focusing on the 3 stresses here that had the biggest effects. We identified the genes that had at least 2 cell types that had an increase in the expression for those stresses, and then cross-reference those across the 3 stresses to identify that there were 6 genes that were upregulated across the 3 different stresses in at least 2 different cell types. And the genes are listed here. And we decided to follow up specifically on SIB1 because it was something that was involved in various cellular processes, including calcium signaling, cell adhesion, apoptosis, which seemed pretty relevant to diabetes, and it hadn't been studied previously in beta cells. So again, going back to our stem cell derived islet system, we use lentiviruses to either knock down or overexpress SIB1. I'm showing the confirmation of that on the top here. We found that knockdown of SIB1 caused dysregulation of insulin release, with there being elevated insulin secretion at both low and high glucose now that we didn't see with the overexpression. This dysregulation of insulin secretion seemed to be because of dysregulation in cytosolic calcium, which was elevated when SIB1 was knocked down. And we also found that cells with SIB1 being knocked down had increased sensitivity to BFA cytokine mix and dapsogorgon treatment by having an upregulation of apoptosis markers under these conditions. We were able to find that overexpression of it would decrease apoptosis under BFA, but didn't see a similar trend for the other stresses that we were testing here. So another thing that we found very surprising, actually, was the endocrine versus the exocrine data that we were able to measure. We weren't certain if we would really get much exocrine in our data set because these were isolated islets, but we had a good amount of exocrine here to allow for us to do a head-to-head comparison. So now we're basically pooling all endocrine together, all exocrine together, and looking at pathway analysis here, and seeing, for example, in the endocrine upregulation and downregulation of various metabolic pathways under BFA stress, or in the cytokine stress here, able to see programmed cell death, PD1 expression, HLA expression, necrosis in the endocrine, the exocrine here. So we're able to see that there are responses here. Tissues were both responding, but both responding in a different way. And so to really get more into what's going on here, we did, so I've been showing you RNA data up to this point. So now we got another patient and did multi-omic data, getting again both ATAC sequencing and RNA sequencing data. This is just showing that we actually did it. Hopefully you guys believe me. We did look at how, for this one, because we couldn't multiplex the conditions, we were just looking at control versus cytokine mix for right now. And looking at, again, the chromatin accessibility of the DNA binding motifs, which we have previously found to be very important when looking at cellular maturation. We're able to, again, see there being cell type specific changes in motif accessibility with cytokine treatment, though one of the things that really stood out to us here was how specific a lot of these changes were in the ductal and acinar, as opposed to a lot of the changes that were being seen in the endocrine cell types were also seen in other endocrine cell types or the exocrine cell types, but maybe not to the greatest extent. So there, again, there are some tissue-specific differences going on here that we don't fully understand yet. However, one of the kind of coolest aspects of the exocrine that we studied here was following up from a result that Kyle Galton's group published a few years ago, where his group identified a single nucleotide polymorphism in a cis regulatory element controlling CFTR expression. He found that was associated with increased risk of type 1 diabetes. Basically, he found a variant in an enhancer that was associated with increased risk of type 1 diabetes. This variant resulted in the enhancer having lower activity, and therefore in ductal cells there was lower CFTR expression that he was able to observe. And so we wanted to see if we could see something similar, but instead of varying the genotype of this enhancer, we wanted to see if cytokine mix would also have a similar effect. And so in the beta cell, again, the beta cell has very little CFTR, and so we don't really see this enhancer being open in the beta cell with or without cytokine treatment. But in the ductal cells, under our PBS control conditions here, we're able to see the enhancer is open. You get the cell cytokine mix, and the enhancer loses accessibility, correlating with what Kyle saw with his genetic risk variant here. And that correlated with a large decrease in the RNA expression of CFTR. So it's not just genetic variants that can do this, but also environmental factors that can do this. So of course, we don't know what the connection mechanistically is of CFTR to increased risk of type 1 diabetes, either genetically or environmentally. So we were obviously very excited about the chromatin data here, and we decided to bring it back to what the beta cells were actually doing. Now looking at RNA, particularly since we looked at the RNA expression of specifically in the beta cell of various chromatin remodelers, and saw different mixtures of these remodelers either having increased expression or decreased expression with the treatment of these various stressors that we were giving them. And so we again did the single cell multiomic sequencing, focus in this case now on BFA versus control, since we saw a greater amount of change in the beta cell with the BFA condition here, and saw this is just a general way of kind of globally showing chromatin changes here, that we had a large number of differentially accessible regions of the DNA that either had increased accessibility or decreased accessibility. And when we looked at specific genes within the beta cell, we found in general that the decreased accessibility was associated with beta cell identity. Here we're showing the insulin gene. You can see kind of here that the peaks are a lot smaller with the BFA treatment than the control. Or for the peaks that typically had increased accessibility, they were associated with a stress response. Again, this is BIP showing now increased accessibility over here of this region of the HSPSA5 or BIP gene. And there's also other genes of kind of unknown significance here that we saw similar increases in the RNA expression in the promoter accessibility. That could probably be a really cool thing to follow up on. But again, looking at other things and trying to get to this, to kind of the core question that we are still motivated by in these sorts of studies which is, what are the beta cells doing? What is special about the beta cells here in terms of their response? And so this is the last piece of data I think I'm going to be showing here. That we did look at both upregulated RNA and upregulated motifs that were specific to the beta cells. In all, we found 43 of them. And looking through that list, the five most interesting ones that we saw here are shown on the screen. So we found that there are five genes associated with apoptosis that specifically had upregulation of RNA, upregulation of motif accessibility that are associated with apoptosis. And this is, this might be part of the core mechanism for what is going on in a beta cell, but to really prove that will require additional studies. So in summary here, we've been able to identify cell type specific responses to various different types of stresses, identifying that some cell types respond more strongly than other cell types, and it's not exclusively just the endocrine cells either. We found common upregulated genes across cell types and endocrine cells. We found tissue-specific pathways, including chromatin remodeling of the T1D risk SNP regulating CF. Actually, that's supposed to be CFTR expression, not CTCF expression. I think about a type of that earlier in the talk as well. That's another gene that we study, but in the context of maturation here. We do see some genetic signatures of beta cell vulnerability here that could be worth following up here. And we did find evidence of modulation of SIB1 and giving the cells pharmaceutical drugs that could be a potential therapeutic target to provide a new diabetes treatment. And so, like I said earlier, this is kind of a long-term strategy of my lab, using this method to study, to understand as much as we can about the biology of diabetes in islets. Part of this is that we have a new preprint out using a similar method, but specifically studying cell type specific responses to Coxsackie virus infection. Which has been associated with the formation of type 1 diabetes as well. So here's the citation to go check out the preprint, and hopefully it'll be published more formally later on this year. So with all that said, I appreciate the invite to share my data with you all. Here's my information about grant support, collaborators, and of course my amazing team here at WashU. And I'm happy to answer any questions you may have. Thank you.
That's thank you so much. That's fantastic. And I really can't, that was one of my questions about Coxsackie. So that's great that you guys have already got that out in the literature. That's really exciting. Here's a question from Michael Kalwat. What do you make of the PP cells having what appeared to be a reduced response to stressors? Was I seeing that correctly?
Yeah, I don't know. I don't understand it. PP cells are not exactly the most popular cell type that is studied in pancreatic islets. Also, another thing as well, and I can't remember if I pointed this out earlier or not, but we actually don't see PP cells in the stem cell derived islets that we produce, and so it's generally been off my radar, except for in the study. I don't know why they would be seemingly resistant, or at least minimally responsive to these stressors, but that could maybe tell us something about the differences in different islets, or so different endocrine cell types to cues that could be interesting to contrast to the beta cells.
This might be a naive question, but do they have CFTR channels?
I'm pretty sure they don't. I didn't look at that specifically. I'm going to stop sharing this so I can see. I don't think we looked at it specifically, but I think we would have noticed it passively if it did have CFTR.
Yeah.
Yeah.
Because they've got that right there. That's a peptide hormone that's involved in digestion. So who knows if they're like marching together with the endocrine pancreas or whatever. And then I asked you a question, I think that was earlier offline. I'm just going to ask it again for everybody else to hear. Like, that's a really interesting finding. The endocrine component of your study was somewhat offline, in response to these cytokines. And can you compare this phenomenon to clinical data sort of surrounding exocrine enzyme secretion in stage 0 to 1 patients, or like their SIBs?
Yeah, I mean, there's been so much clinical data collected that is mostly divorced from the molecular data that we get here with sequencing. I would be very interested in seeing, really understanding more about patient like clinical readouts and patient heterogeneity and how that interacts here. If we could see, and again, the CFTR is maybe one of the more easier examples here, but if we could see like differences in CFTR activity that correlate with these clinical markers of exocrine activity or autoimmunity or something. I think it could really help, like really having a concentrated effort to combine the clinical data with what we can do in the laboratory. I think would be a major advancement in our understanding for type 1 diabetes pathology.
Amen. That is the truth right there. And here's another question I had for you. Can we just kind of go back to that figure 3A? Like, can you discuss your findings regarding the upright, sort of a nod to, the buzz in the field of the primary cilia these days? And Jing Hughes at Wash U is very involved in that, and Matt Marins.
Mm.
Um, can you talk a little bit about your findings regarding upregulated ciliary landscape RNA in the endocrine cells under BFA conditions? Um,
Yeah,
it's
I
kind of
mean,
because it's like the smallest amount of change in that figure, but
mm.
because it's like the percent of Golgi affected sort of by the BFA that's targeted to the primary cilia is low compared to overall percent of Golgi in the cell. Could that be like masking it, or I don't know?
Oh, Dr. Hughes is down the hall. I can get her to answer that question for you. I don't know if her lab and my lab are right beside each other, so I'm gonna not answer this as good as she would. I mean, obviously the cilia is amazingly important for beta cell function and probably also the pathology of the cells. I wouldn't be surprised. In general, it works as a signaling hub as well.
Yeah.
And one of the things that I personally have been very curious about has been, is one of the differences across these endocrine cell types here, differences in the primary cilia that
Yeah.
perhaps because of the signaling that occurs there that might express why there is, this is all speculation, I don't have any data for this, but I wouldn't be surprised if there is differences in either primary cilia presentation or the signaling receptors that are present on the primary cilia across different cell types. Perhaps the beta cells have something special about them beyond their presence that could be causing a lot of downstream effects. And how you would even study that, you would need to ask Gene Hughes.
Yeah, no, she's definitely the expert there, but it is we're kind of following that story closely because it's very interesting and it's like late breaking news all the time on it.
Mm.
But and there's this dopaminergic receptors in the ciliary membranes, and it would be cool to figure out whether or not the stem cell derived entities or cells also have the same ciliary phenotype.
Mm.
So
Well, I can say that that is something that we have looked at. I can't say what that is yet because we're trying to come up with trying to finalize a story around that.
great.
So stay
Yep.
tuned.
We will say stay tuned. That's exciting. Okay, well, thank you so much. I appreciate everyone going a little over, but it was definitely worth it to hear this story in full. And thanks again for all the great work that's going on in your lab and WashU in general. Really some interesting work coming out of WashU. It's fantastic. So have a great rest of your day.
Yeah, you too. Thank you for the invite.
Thanks. Bye-bye.
Okay, bye bye.